This is part 2 of the walkthrough of the AQA GCSE Biology 2022 Higher Tier Paper 2. Question 6. Energy flows through an ecosystem and materials are recycled. Figure 6 shows the water cycle. 6.1. Name process X. So process X is showing us an arrow going from water to water in the air. So what that must be, if it's moving from water as a liquid into the air, which is going to be water vapor, so this would actually be our vapor, this process must be evaporation. Name the process by which water is absorbed into plant roots. So water going into roots, that's knowledge of cell biology now, that's osmosis. But 6.3 now give 2 uses of water in plants. So how would the water that's gotten into the roots be used by the plant? So one use of water in plants, again thinking of paper 1 now, it's got to be photosynthesis, carbon dioxide and water producing glucose and oxygen. But another reason why plants would use water is that they can use the water to transport substances. And one thing that they would probably transport is what is produced in photosynthesis, which is a glucose, or sugars. And that movement of sugar in the plant is called translocation. Figure 7 shows the flow of energy through a food chain. The numbers are in kilojoules per meter squared per year. So all of these values have this unit. The cow is more efficient than the grass at converting energy. The energy conversion efficiency of the cow is 4.098%. Calculate how many times more efficient the cow is at converting energy than the grass. The equation for energy conversion efficiency is here. Give your answer to 3 significant figures. Right. So we've got to use this equation. However, it's asking us to calculate how many times more. So that means that we need 2 values of efficiency, and then we need to find out a factor of how many times more. So that means that we don't need in our final answer a percentage. We need how many more times. And that's why it says number of times. So they've given us the energy efficiency of the cow already. So we then need the energy efficiency of the grass. That's what we need to find out. So energy efficiency of grass. This is what we need. So for us to find that out, we need to find the energy used for growth, and we need the energy input, and then we need to multiply that by 100. So going back to our figure now, energy used for growth and energy input for grass. So here is our growth for grass, 21,800. There is our input, 1.7 million. So we would just need to plug that into our equation then. So 21,800 divided by 1.7 million. And then we need to times that by 100, and then we should be able to get our energy efficiency from there. So plugging that into our calculator now, we should come out with an answer of 1.28 It's best that we keep our efficiency at 3 significant figures, so it makes it easier for us to multiply to find the number of times. So again, our efficiency for our cow is 4.098%. So all we're doing then is doing 4.098 divided by 1.28, and that will give us our answer for number of times. And our value that we get is 3.19921875. So putting that to 3 significant figures, that would be 3.20. 6.5. It is more energy efficient to rear cows indoors than to rear cows outdoors. Give 2 reasons why. So we're thinking then in terms of converting the energy from our input to growth is going to be more efficient doing that for cows indoors than outdoors. So what might be the reasons then? Well, if our cows are indoors, they're going to be in a certain space. So our cows can be in a certain area now, as opposed to outdoors where they're going to be more spread out. Well, what that means then is that the cows, when they're indoors, they have less space to move. So they have a limited area in which they can move. Why is that useful? It means that they're going to be using less energy. So less energy is going to be used for movement. So then if less energy is used for movement, more energy is going to be used for growth. The second reason, if these cows are kept indoors, the thing that will happen then is that because they're closer to each other, they'll be able to keep each other warm. So any heat that's lost from one cow will be transferred to another nearby cow. So that means then that they'll be able to use less energy for heat. And those are the 2 reasons. If you think about why energy is important, there are 3 reasons why. One is for movement, one is for keeping warm, and one is for chemical reactions. Well, the chemical reactions is the one that we want because we want to get more growth from the cow, so we want less of these to be occurring. 6.6 suggests 2 possible disadvantages of rearing cows indoors. Well, a disadvantage of rearing cows indoors now, if we're thinking about these cows here, what could be a problem is if this cow develops a disease, because they're so near to each other by being indoors, that could mean that the disease could spread to this cow, and then it could spread to another cow, and then another cow, and another cow. So that means that the chances of spreading a disease is going to be greater. And that's of course something that you definitely don't want, because if it's some crops or some animal that you want for meat, the chances of spreading a disease could affect the rest of your population. So it would increase the spread of disease. The other problem with rearing the cows indoors or rearing any other animal indoors is that they're not supposed to be indoors. They're supposed to be out in the open where they're able to move around freely. And if you lock an animal in a cage or in a certain space, their behavior will change. So they're going to be more aggressive and they might start causing more damage. So disadvantage now is that you'd get a change in behavior of your animal, or in this case the cow. And what that change in behavior will be is to be more aggressive, and then it'll be harder to manage. Question 7. A scientist found a polluted pond which had a new type of blue algae in the water. The blue color of the algae was caused by a mutation. 7.1. What is a mutation? Well, this is a recall of the definition of mutation now. It's just a change in base sequence of DNA. I think it is actually even fine if we said a change in DNA. The scientists measured the number of blue algal cells in a sample of the pond water. The scientists used a special slide which has a counting grid. This is the method used. So diluting some pond water to a volume of 10 centimeter cubed with some distilled water. They're placing a drop of the diluted water on the special slide, which is shown here. They place a thick cover slip over this diluted pond water, and that gives a depth of 0.1 millimeters. And then they're using a microscope to count the number of algal cells in 0.2 millimeter by 0.2 millimeter square on the counting grid. Okay, so they've shown us the special slide that they've used. And figure 9 is showing the counting grid. So, as I said, area 0.2 by 0.2 millimeters. 7.2. How many algal cells are in the 0.2 by 0.2 millimeter square in figure 9? Use the following procedure. Count all cells that are completely within the 0.2 millimeter by 0.2 millimeter square in the counting grid. Count cells that are touching the left side or the lower side of the square. Do not count cells that are touching the right side or the top side of the square. Right, so we have to only use left side and lower side when we are counting. So what we can do now is we can just highlight the ones that we should not be counting because these ones are not completely in the grid. So that 1, 2, 3, 4, 5, 6, 7. Okay, and 8 because it's not completely inside. Right, so we can also not use this one, that one, and this one here. Okay, so let's begin our count then. So left and lower, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16. So our count should be 16. 7.3. 1 week later, the scientists repeated the test and counted 14 cells on the 0.2 by 0.2 millimeter counting grid. Calculate the number of algal cells in 1 millimeter cubed of undiluted pond water. Use the scientist's second count of 14 cells. So 1 millimeter cubed undiluted pond water, we need to find out the number of cells in that. So you've put in bold undiluted. So we have in our experiment at the top here, we've got diluted pond water. Remember that they diluted 2.5 in 10 centimeters to a volume of 10 centimeters. Okay, then let's go and check that then. So our undiluted, we need to get that from diluted. Checking again now, it's 1 millimeter cubed. But we're giving a volume also. So our volume that we're given, because we're given lots of dimensions here, we're giving a depth of 0.1 millimeters, but we're also given an area 0.2 by 0.2. So we're given 0.2 by 0.2. So we can get our volume from there. So in simple terms, what we've got now in our question before 7.3, we had 16 cells in, well, it was 2.5 centimeters cubed in 10 up to a volume of 10. So realistically, it was 10 centimeters, which is the diluted anyway. But then that was also in a volume of 0.2 times 0.2 times 0.1. So that should be volume of 0.004, 0.004 millimeters cubed. So we need to get that to this situation now, which is 1 millimeter cubed undiluted. Now, remember, they've asked us to actually use 14 cells, so we've got to change this now to 14. So what we want now is how many cells in 1 millimeter cubed, but we also need our actual undiluted. So we want 2.5 centimeters cubed. So what we're going to do now is we're going to do some conversions here. We want to find out the factor to get from 0.004 to 1, and we want the factor to go from 10 to 2.5, and then we'll be able to get our answer. So let's start with 14 cells in 0.04. How many would that be in 1 millimeter? So what we'd have to do is find out how do we get from 0.004 to 1. So we just do 1 divided by 0.004, and that will be a factor of 250 times. So times 250. So if we do that in terms of our cells now, we would do 14 multiplied by 250, and that would give us 3,500. So we now know that we've got 3,500 cells in 2.5 centimeters cubed, but in 1 millimeter. So what we need to find out now finally then is how much would that be in undiluted. So what we had actually done is we'd had 2.5 centimeters and then we had diluted that to 10. So what we've done then is we've actually found our factor right there, 10 divided by 2.5, and that would be 4. So we've diluted by a factor of 4. So what we need to do to find out what it would be undiluted is actually take into account this factor in our number of cells. So what we need to do then is 3,500 multiplied by 4, and then that would give us our complete number of cells, which is 14,000. Seven 7.4. Suggest why the scientist diluted the pond water before placing it on the special slide. Well, look at the special slide. There's our counting grid, and we obviously counted 16 cells when we did it, when we're counting from the grid. However, if it was undiluted, it would actually be near this number, 14,000. So obviously that would be a lot more difficult to count. So by diluting it, it's making it easier to count. (7.5). A student repeated the scientist's method. The student used a thin cover slip over the diluted pond water instead of the thick cover slip. The liquid pulled the thin cover slip downwards slightly. Explain how the use of the thin cover slip would affect the results for the cell count. So it says that the liquid pulled the thin cover slip downwards slightly. So let's try and visualize that, because they've given us the picture here. So this cover slip would be a bit thinner, and then it said that the liquid pulled the cover slip down slightly. So essentially, the water is actually being pushed down by the fact that the cover slip is moving down. So what that's actually doing is going to kind of compress and squash the water down. Now, if that's doing that, then that means that this depth is going to change. Because if the pond water is actually further down than it should be, that means the depth should be lower. So what's actually going to happen then is that these cells would appear lower, and then it might mean that they're going to be a bit more squashed. Now, what might that mean if they're a bit more squashed? That might mean that some of these cells, at least these ones that are a bit closer to each other, they might actually be on top of each other. And then when it comes to counting, the cells might actually look like one cell instead of it being two cells. So what I think that might mean then is that the count, because it's asking us how would it affect the results of the cell count, the count would be lower than expected, or lower than it actually should be. And the reason is that the depth would be lower, and then cells would be squashed and on top of each other. Question 8. An echidna is a mammal that lives in Australia. Figure 10 shows an echidna. Figure 11 shows how the body temperature of the echidna varies in warm weather and in cold weather. Okay, so here's our graph showing us these two different weather patterns. Figure 12 shows how body temperature varies. So there's a body temperature of a human, yes. 8.1. Compare the variation in body temperature of the echidna in warm weather with the variation in body temperature of the human. Use data from figure 11 and figure 12. So we want the variation in body temperature of the echidna in warm weather with the variation in body temperature of the human. So we're comparing. So we need to be using words such as greater, less, more. Have to be using that. But it says use data from figure 11 and figure 12. So we want to see how this variation is different. So let's start with the echidna in warm weather then. So we're looking at how it varies. So the temperature starts at 30 degrees. The best thing for us to do next is to see where it ends, because that gives us an idea of variation. So it ends just there, and using the scale, that is going to be 30 to 33 degrees. So it ends at 33 degrees there. The other way that we can compare the variation is by seeing how much it goes up by in terms of its greatest increase and its greatest decrease. So you can see that the greatest increase is just here now, and that is 35, halfway. And the lowest body temperature is here, which appears to be 27. So we could say two things then. That overall it increases just by 3 degrees. So overall increase is 3 degrees plus 3 degrees. But it goes from, in terms of its range then, because the lowest to highest is range, 27 to 35 degrees C for range. A final thing that we could actually do here is we could see how many times it goes up and down. This up and down is the fluctuations of the temperature. So you can see there's 1, 2, 3, 4, 5, 6, 7, 8, 9 fluctuations there. So what we can do is think about that as well. Maybe just make a note of it here. And then we can do the same thing for our human. So the first thing that we did was we looked at the start. The start's at 37, and that should be expected because that's human body temperature. 37, and it ends just below 37. Now let's look at the scale here. Can you see that the scale is a bit smaller in terms of its variation compared to our echidna now? Because this is 36, 37, 38, so we're really only dealing with decimal places in terms of its increase right now. So this end point at 10 weeks is 36.9. So it means that overall it's only changed at its end by 0.1 degree C. Let's look at our ranges then. So our lowest temperature, this point here, and that is 36.2. Our highest point, you can see, is this, which is 37.2. So that means the range is 36.2 to 37.2. So you can see there's a smaller range then. Much smaller range. We could talk about the fluctuations then. Fluctuations from 37. Well, the changes are actually less because the range is smaller. So another thing that we could say then is that there are less fluctuations. Okay. So let's put that in our answer then. I think the most important things that we can take from here are the fact that the range is so different. And then we said there are less fluctuations. So let's put that in our answer here. So for the echidna, range from, let's just make sure we got the right values, 27 to 35. And human from 36.2 to 37.2. So, which has a wider range, is the echidna. And then we said that for the human, it has less fluctuations of this temperature. In cold winter months, the echidna hibernates. During hibernation, the echidna's body temperature decreases to below 5 degrees. The echidna sleeps for up to 17 days at a time. The echidna's rate of metabolism slows down. 8.2. Explain why the decrease in body temperature is an advantage to the echidna during hibernation. So there's this drop in the body temperature, and that is going to be an advantage to this echidna. So let's see here. It tells us about the temperature dropping. It's sleeping for up to 17 days at a time, but the rate of metabolism slows down. Now, this is an important thing here because metabolism is the rate of chemical reactions in the body. Now, if the echidna is hibernating, what the echidna doesn't want to do is die. So, what that means that the echidna is going to need to do is it's going to need to use its energy to keep itself alive. But the echidna, if it's hibernating, is not actually eating. So that means it's got to get its food from whatever stores that it has. So, for doing that up to 17 days at a time, it needs to have this metabolism slowing down. If the metabolism was really fast and it was sleeping up to 17 days at a time, then all the energy that it had stored will be used very quickly, and then it wouldn't be able to survive. So, having these chemical reactions slow down, and examples of chemical reactions are respiration, but also some examples of digestion are metabolic reactions. So having these occurring less or slower means that it's able to keep using the food stores that it has for a longer period of time. So what we can say then is stores of energy last for a longer period of time. I think we also need to try and make a link to this decrease in body temperature now. So what would be the decrease in body temperature link? Well, it's going to mean that there's going to be less energy that's going to be lost from the body. And of course, if there's less energy that's lost from the body in terms of heat, then that means more of it can be used to keep the echidna alive. 8.3. During hibernation, the echidna wakes up several times. Each time the echidna wakes up, it becomes active and its body temperature increases to over 30 degrees. Explain why the echidna has a higher body temperature when it is active. Right, so talking about the opposite really now. When it's active, body temperature is really, really high. So what we've got to think about now is, what does this activity mean? Well, if it's active, it probably means that it's going to be moving. And if it's moving, that is respiration that's being carried out. And we should always remember that respiration is going to produce heat. So that should be our link there. When active, it is moving, which requires energy from respiration. And then this energy, as we said, that's coming from respiration, it means that more of it then is going to be releasing this heat. So the next point that we probably want to say is respiration is going to release heat. Okay, 8.4. An echidna can dilate and constrict blood vessels in its skin. Explain how the dilation of blood vessels in the skin can help decrease the body temperature. So dilation, with it's talking about vasodilation now. So this is control of body temperature. So how is this dilation of blood vessels going to help decrease the body temperature? Well, if this is the surface of the skin of the echidna, skin surface here, then below this skin surface, we are going to find blood vessels. Now, these blood vessels are of course coming from the rest of the body now, but what will happen is these blood vessels, especially down here, these are able to either dilate, so get bigger, or they're able to constrict, get smaller. So if the blood vessels are dilated, that would mean then that more of the blood is flowing to the surface of the skin. So that will just be increased. So we've got more blood flowing in these blood vessels. This is close to the surface of the skin. How is that going to link to body temperature now? Well, the blood is going to be carrying this heat. So if there's more blood close to the surface of the skin, that means that the blood is going to be carrying the heat. So if it's closer to the skin surface, then it's going to be easier for the heat to be lost. So the heat will just travel through the surface of the skin and then out of the body. So what will that do then? That will cause the temperature to decrease, and that's just going to cool the echidna down. So we want to try and put all of those points now in our answer. So first thing we're saying now is, what is the dilation going to do? So the dilation is going to mean that more blood travels close to the surface of the skin. And then the blood is carrying heat, showing us heat energy. So that means that more heat is lost. And then, as we said, then if more heat is lost from this echidna, then it's going to be cooled down. An athlete trained in a hot climate. The athlete lost a large volume of water each day in sweat. An athlete's energy intake each day from food was 20,000 kilojoules. Evaporation of 1 centimeter cubed of sweat requires 2.5 kilojoules of energy. 40% of the athlete's daily energy intake was used to evaporate sweat. Calculate the volume of sweat the athlete lost each day. Give your answer in decimeters cubed. And they've helped us by giving us the conversion here, that 1 decimeter cubed is equal to 1,000 centimeters cubed. So volume of sweat lost in 1 day we need to actually calculate. Well, they've told us that the intake of energy from food is 20,000. So this is the total energy that is taken in from food. But 40% of this is used to evaporate sweat. So we need to find 40% of 20,000 before we can go further, and that will then help us calculate our final answer eventually. So 40% of 20,000 is going to be 8,000 kilojoules. Okay, so we know now that 8,000 kilojoules is going to be lost from that as sweat. But it's told us now that 1 centimeter cubed is equivalent to 2.5 kilojoules of energy. So 1 centimeter cubed that's evaporated is equal to 2.5 kilojoules of energy. So we want then to find out what's the volume when 8,000 kilojoules is lost. So again, we're finding this factor now. So what we need to do is do 8,000 divided by 2.5, and we'd get 3,200 kilojoules. Okay. So we then know, using that same factor, if it's 1 centimeter cubed to 2.5 kilojoules, then that means that we've got 3,200 centimeter cubed. That will be our volume of sweat lost. However, we need to convert to decimeter cubed. So those 200 divided by 1,000 would be equal to 3.2 decimeter cubed. 8.6 suggests why the athlete was advised to take salt tablets each day. Well, what's happening? They're doing their exercise, losing water as part of sweat. But what will the water that's in this sweat also contain? It will contain ions. So the ions is what will be part of the sweat. So what we need to then say is that the salt tablets are going to replace the ions that are lost in the sweat. Remember that these ions that we're really talking about is going to be sodium, chloride, potassium. These are, specific ions that you need, which can get lost in the sweat. Question 9. Students investigated the response of plant shoots to one-sided light. Figure 13 shows how the students set up 3 experiments. So experiment A, you've got a seed, and then the shoot is grown from it. They've put ink marks on the shoot, and then light is from one side, and then left that for 12 hours, and then they've seen a change. Experiment B, seed with a shoot, again ink marks, but they've put in a light-proof box, left for the same amount of time. And then experiment C, no box, but light in all directions, and they've left it again for the same period of time and seen the results. So let's look at the results though after 12 hours for each of the experiments before we look at the next question. So in experiment A, the shoot has bent towards the light. So this is our example of our phototropism, because it's the response of the plant due to light. In the box, we're seeing there's growth, but there's no bending towards any light. It's just grown in a uniform direction upwards. And then the same thing has happened in experiment C. But in this case, it's due to the light coming from all directions. So no light here. Light one side. And then light in all sides. Okay. So 9.1, suggest 2 control variables the students should have used in their investigation. So something that needs to be the same, kept the same in each of those experiments. Well, I don't think we can talk about the time because that was already mentioned. 12 hours, 12 hours, 12 hours. So it's got to be other things now. So think about what, if it was to change, could affect the results seen here. Well, a thing that has to be kept the same is what type of seed were they using? So the seed's got to come from some type of plant. So the type or the species of plant should be kept the same. So type of plant definitely need to keep the same. What else could affect the results seen here? Well, what's going to influence how well the shoot grows? Well, it's going to be some of the factors such as temperature. So you only want to give 2, but of course other ones may be the amount of water given. So volume of water could be that. 9.2. Describe how experiment B and experiment C acted as controls for the investigation. So remember, there's a difference between control and control variable. In this question, they try to make it very clear by putting them together now. So control variable, what you're keeping the same in the experiment, but control is what is going to be put in the experiment to know for sure that your results are as a direct result of the independent variable. So in this case, the independent variable is the direction of the light. So here, of course, in A, one side, C, all sides, B, no sides. So why did they put B here? Why did they set up C? So what is B trying to show? What is C trying to show? So really, the main reason why we use control is for comparison. So what are we trying to compare in B? Well, we're trying to compare no light in B compared to light from one side in A. That's really what we're trying to show. So the comparison between no light and one-sided light. But in C, what we're then comparing is one-sided light to light from all sides. 9.3. Give 2 conclusions that the students could make from the ink marks on the chute in experiment A. So looking at the ink marks now, what is that telling us? So let's look at chute A. What do we notice about the ink marks? Well, at the bottom of the chute, you can see that the ink marks are very close together. But as you move upwards towards the tip of the chute, the ink marks are more separate now. So what is that telling us then? Well, you have to think about the reason of this chute growing. The reason why the chute is growing is because cells are dividing or the cells are elongating. So there's either going to be cell division or cell elongation that's going to be happening. But because there are greater separations or greater distance between the ink marks, that's telling us then that this cell division is occurring towards the top of the chute or the tip of the chute. So it's not really occurring at the bottom of the chute. So first things that we can say then is that the cell division is occurring at the tip of the chute. Okay. We need to give 2 conclusions here. So what could be a second conclusion that we can get from here now? Well, remember, what's happening is the shoot is bending towards the light. So, if the shoot is bending towards the light, what do we also notice then? That this bending, and therefore the cell division or cell elongation, is actually occurring on the side away from the light. Because if it was occurring on both sides, then the shoot would grow straight upwards, as shown in B and C. So what we've got to say then from there is that the cell division is occurring most away from the light. 9.4. Name the type of response shown by the seedling in experiment A. Well, as we said before, what this investigation is showing is phototropism. Remember that photo means light and tropism is a response to a stimulus. So we're looking at phototropism here. Specifically, because in experiment A, the shoot is growing to the direction of the light, you actually call it positive phototropism. But we only need to show that it's phototropism here. If it was in response to gravitational field, then it would be gravitropism. Auxin is a plant hormone. Auxin is made in the shoot tip. Scientists investigated the role of auxin in the response of shoot tips to light. This is the method used. Grow 4 seedlings in the dark for a few days. Cut the tip off the shoot of each seedling. Place each shoot tip on a small block of agar jelly. Place the shoot tips and agar in different conditions, as shown in figure 14. After 24 hours, measure the mass of auxin in the agar blocks. So we've got figure 14 here, D, E, F, and G now. So what does it tell us? D kept in the dark. And then we've got this value here, which is telling us the mass of auxin. E kept in the dark, but a glass block was put between our agar. And then we can see that the masses are relatively equal on both sides. Light shining on one side, and we can see again the glass is put in between the block. We've got more of the auxin on the side away from the light. And then G, glass is now put in between the shoot tip. Light on one side again, and then we've got equal amounts of auxin on both sides. The numbers on each block show the mass of auxin that diffused into the blocks from the shoot tips. The mass of auxin is given in arbitrary units. 9.5. A scientist made a hypothesis. Light causes auxin to move from the side of the shoot nearest to the light to the side furthest from the light. Describe the evidence from figure 14 which supports the hypothesis. So, light causing auxin to move from the side of the shoot nearest to the light to the side furthest from the light. Is there any evidence to support that? So, from nearest to furthest. Okay, so let's just make a note. Nearest to furthest is their hypothesis. Well, do we have anything to support that? Well, if we look at E, there is no light. Almost equal amounts of auxin on both sides. F, same situation as E in terms of glass separating the block. However, light on one side now causes movement, possibly, of the auxin onto the side furthest from the light. So by looking at E and F and comparing them, that can give us some support to the hypothesis nearest to furthest. But let's look at G. Well, G has light on one side, but the glass is completely blocking the agar and the tip of the shoe. So maybe this G can actually support this as well, because it then might suggest that the auxin, because the auxin is present in the shoe tip, is not able to diffuse to this side here because the glass is blocking it from happening. So I think we can use E, F, and G to support this hypothesis now. So what we can say is in E, there's similar amounts of auxin. However, in F, when the light is shining on one side, there is more mass of auxin on the side furthest from the light. In G, even though there's one-sided light, because the glass is there, it's preventing the auxin from diffusing to the side furthest from the light. That should be good enough for our 3 points. So let's say again, E has similar masses of auxin, but we have to say there's no light present. But in F, when one-sided light is added, more mass of auxin on the side furthest from light. And then again, G can support that even further. In G, G has one-sided light, but the glass block prevents auxin diffusing to side furthest from light. 9.6. Another scientist made a different hypothesis. Light causes the breakdown of auxin. Give the evidence from figure 14 that shows that auxin is not broken down by light. So auxin not broken down by light now. So this is the hypothesis, auxin broken down by light. So what cannot support this? Right, so in dark, with the glass block there, we've got 12.5, 12.4, again similar. But in the light here, there is less auxin on the side facing the light. Now, I think if the light was breaking down the auxin, then we wouldn't get more on the side furthest from the light. So we would still have something similar to 12.5 on this side, but then we'd have something lower than 12.4 on this side. So that doesn't support it. But if we look at G, G has 12.6, which is similar to our E. Remember, E has no light. So if the light was breaking down the auxin, then this value should definitely be lower. So what we can say then is that the mass of auxin in E and G are similar, because F would support it if this value was not higher. So I think we can say that the masses of E and G are similar, even though G is in the light and E is not.